Hi everyone, my name is Precious, and for my projects taking place at Holland College in the Bioscience Technology Department, I explore the potential effect of strawberry extract and ellagic acid in MTA MB231 breast cancer cells. And as we all know, breast cancer remains a major global health burden, with triple negative breast cancer representing one of the most aggressive subtypes. Natural compounds such as ellagic acid, which is a polyphenol abundant in strawberries, have demonstrated antioxidant and anti-cancer potential, and due to its aggressive nature and resistance, the MDA-MB231 cells serve as a well-established model for the study. Therefore, building on that, I hypothesized that ellagic acid extracted from strawberries would induce ROS-dependent apoptotic morphological changes in MDA-MB231 breast cancer cells, leading to decreased cell viability and reduced invasiveness of breast cancer. So to begin, I started my experiment by extracting ellagic acid from strawberries. Initially, I freeze-dried my strawberries, then conducted homogenization, and then the mixed mode solid phase extraction process. Afterwards, I conducted my UPLC analysis to further confirm that ellagic acid was actually present in my extract and to quantify the amount present in my extract as well. And as you can see at figure 1, the UPLC chromatogram shows a peak of ellagic acid which matches the ellagic acid standard, further confirming the presence of the compound in the extract. However, the concentration was only really low, so it was too low to be used for further cell-based assays. So, for all my biological experiments, I moved forward with pure ellagic acid compound instead of both the extract and the compound. Cell viability was assessed using the MTT assay by treating the MDA-MB231 cells with ellagic acid at concentrations ranging from 1 micromolar to 40 micromolar across 24, 48, and 72-hour time points. The cell viability results showed that there was no significant effect at 24 hours. At 48 hours, a significant decrease in cell viability was observed at 40 micromolar, showing higher concentrations of ellagic acid had cytotoxic effects. And at 72 hours, we can see in figure 2 that there could have possibly been a common metabolic frustration due to a significant increase in cell viability at lower doses. However, at the same time point, a significant cytotoxic threshold was reached at 30, 35, and 40 micromolar. Now, after the identification of these optimal doses of ellagic acid to be used for further experiments, to further confirm the motive mechanism behind cytotoxic effects, I looked at the reactive oxygen species using the DCHFDA assay. The ROS results showed that ellagic acid caused changes in reactive oxygen species levels across multiple concentrations and time points. At 24 hours, as you can see in figure 3, there was an early variable oxidative stress response. The IS-ROS elevation was observed at 40 micromolar after 48 hours, which matched the time point where reduced variability was also seen in the MTT assay. This suggests that ellagic acid may have disturbed the redox balance of the cancer cells, further contributing to the oxidative stress and cell damage. And to further support this idea, post 72 hours apoptosis analysis, apoptosis cell counts increased significantly with increasing concentration. From the selected concentrations based on the MTT assay, the untreated control had about 6.67 apoptotic cells, while the numbers increased to 16, 23, and 33 at 30, 35, and 40 micromolar concentrations, respectively. This shows a clear dose-dependent induction of apoptosis by the ellagic acid compound. So Putting all of this together, ellagic acid appears to reduce cell viability and also disrupt oxidative balance and induce apoptosis in these breast cancer cells. And while there was some initial increase in viability at lower concentrations, the overall effect at higher doses and longer exposure was clearly or is clearly cytotoxic. In terms of limitations, the main one was a low yield of ellagic acid from the strawberry extraction process, which prevented direct comparison between the crude extract and the purified compound. So moving forward, future work should focus on improving extraction methods as well as exploring the underlying molecular pathways, especially those related to oxidative stress and apoptosis. It would be very useful to test this in additional cancer models or even in vivo systems. Overall, these findings suggest that ellagic acid has promising potential as a natural anti-cancer agent, particularly for aggressive breast cancer subtypes.